In this lecture, we're going to be covering the last part of Chapter 13 and discussing how a change in DNA can become a mutation. If you remember from our previous lecture, DNA becomes RNA, which eventually yields a protein. So, if you start within the nucleus and you've got the DNA being transcribed into your mRNA, then your mRNA is processed, leaves, and is read by the ribosomes. The tRNA bring in the amino acids, and a polypeptide is eventually formed in a process called translation. Once that polypeptide is folded, that's what we call a protein. Now, if you remember, proteins have lots of different jobs. Some proteins form the keratin in your hair or the collagen in your skin, and some proteins act as factories. But if one of those factories or one of those proteins that forms the collagen in your skin isn't formed correctly, and then your cells continue to pass on bad code to the next generation, you're going to have a problem because those proteins are very, very important for your efficient running of your body. So the picture on the top right is a picture of ATP synthase. ATP synthase is a very complicated protein, but we've heard of it before. If you remember, ATP synthase uses the flow of hydrogen ions to produce ATP, hence the name ATP synthase. So it's coded for in your DNA. You don't want the code in your DNA to have a mistake, or else the ATP synthase, which you can see is very complicated. It has lots of little different pieces. And if even just one of those pieces, like say right there, gets broken, the whole protein isn't going to work. Just like any machine where one piece of it is broken doesn't work. When my car had just a little tiny rubber belt break, the engine wouldn't run. And so proteins are very similar. If there is a break in this code right here in the DNA molecule, then when the RNA molecule is made, that will be broken too, and then when the protein is made, that will also be broken. So DNA has to be what we call conserved or protected, which is why it's so very, very careful when it replicates itself so that a mistake isn't made. Because remember, DNA makes RNA, makes protein, and that makes you live. So you might think, well, if there is one tiny error in my DNA, that's going to cause a big problem. And sometimes it does. But most of the time it's okay because there is a redundancy in the code to make each amino acid. So if something's redundant, it happens again and again and again. Right? Well, redundancy in code means that you have lots of different codes that give you the same outcome. So let's look right here at leucine. So leucine can be made from CUU, CUC, CUA, or CUG. So those are all redundant codes for the same amino acid, which means if there's a mistake in the DNA code for that last piece of the transcript, it's not going to cause a problem. You're going to be just fine. We're not going to see that at all. You'll still end up with leucine in the amino acid, and the protein will still work perfectly. On this slide, you're going to see lots of different types of mutations, and we're going to go over each one in turn. If you look here, we've got our original template strand. If this nucleotide was changed to an A instead of the G, we would end up with a U in our transcript. But because of that redundancy that I talked about last time, the GGU codes for glycine just like GGC does. So we won't even see that mutation. It still makes the same amino acid, and the mutation is what we call a silent mutation. In this case, a T is substituted instead of a C, and so we end up with an A on our transcript, which will give us one different amino acid. Okay, so we have a new sequence of amino acids. It might disturb the protein a little bit, or the protein could be just fine. Hard to say. Okay, in this case, we switch an A from a T, and I end up with a UAG here, which is a stop codon. This would be very, very bad because now it doesn't make the whole protein at all. It stops right after the start codon. So in that case, that one, what we call a point mutation, or one change at one point in the DNA molecule, is going to cause a very big difference. So you can see here, you can have one change and it makes no difference, a small difference, or a big difference. It just depends on where that point mutation occurs. A frame shift mutation, on the other hand, is when you usually delete or add a nucleotide in the DNA molecule. So it's actually not added on, or you accidentally add an extra. What that does is that changes all of the codons for the rest of it. I'm going to show you in a second a better example, okay, of some different frame shift mutations. However, first I wanted to review the point mutation. Remember, a point mutation happens at one point in the DNA molecule, which makes a change at one point in the mRNA molecule, and then can change one base or possibly no bases. So if I was to look at the sentence above, the cat ate the rat, I could change one letter in this sentence and it would end up the bat ate the rat. Now, in general, you've got a carnivore eating a rodent, okay? So it hasn't changed it that much. Maybe, depending on your story, the change might be really super important or not important at all, okay? So in this case, it wouldn't be important at all. In this case, made a slight change, but not really important. However, as I told you, sometimes a point mutation can be really, really bad. So in this case, we've got wild-type hemoglobin DNA. So hemoglobin is in your red blood cells, and it helps carry your oxygen in your red blood cells. So you can imagine that if hemoglobin was messed up, you'd have a hard time running because you wouldn't be getting as much oxygen, right? And it might make you sick, and in some severe cases, you could even die. Well, some people have a point mutation where this T was transferred to an A, right? Which means that I'm going to have a change in my mRNA, which changes just one amino acid in the entire protein. It goes from a glutamine to a valine. Now, is this good or is this bad? Strangely, it's a little bit of both. What happens with the abnormal hemoglobin is it crystallizes and it changes the shape of the red blood cell. So not only is it this weird and funky shape, but it doesn't carry oxygen as efficiently, okay? It also has a tendency to get clotted in your blood vessels. And so people who have sickle cell anemia have a problem getting oxygen to the rest of their body. They're not healthy for all of their lives and have a tendency to die young. However, red blood cells are easily infected by malaria. So, however, sickle cells are not easily infected with malaria. So in tropical areas, having sickle cell anemia is a good thing to a certain extent, right? How about if you were heterozygous and you had both healthy blood cells and not healthy blood cells, okay? That actually allows you to be immune to malaria, but at the same time have healthy red blood cells that keep your body healthy and efficient. So people who are heterozygous for the mutation are resistant to malaria, but usually healthy. People who are homozygous dominant have healthy cells, but they're not resistant to malaria. So sickle cell anemia is actually really common in the tropics, where malaria is also common, because people who have sickle cell anemia are protected. In general, frame shift mutations can be really bad. And the reason why is because once you change one mutation, you either delete or add a nucleotide into the sequence, it changes all of the codons after that. So remember, like the cat ate the rat. We're reading in 3-letter words, right? Each word tells us something. So mRNA is also read in those 3-letter increments called codons, if you remember correctly. So those codons are read in 3 letters. So if I took out one letter, right here, I took out the E, now it changes every word in my sentence. When that happens, that means that that entire protein can't be made. In this case, the deer is not able to make the pigment protein necessary for it to be a normal deer. That was most likely a frameshift mutation because it wasn't just a broken protein. That protein wasn't made at all. I told you that our DNA is really good about replicating itself, and it checks itself to make sure no mistakes are made. It is very good, but like anything, it's not perfect, and mistakes are made naturally. However, there are some chemicals that cause DNA to make mistakes more often. Those things can be UV sunlight, X-rays, different carcinogens, preservatives in your food, cleaning products, even viruses and bacteria can sometimes cause mutations. Anything that causes an increased rate in mutation is called a mutagen. Now, you may have heard that there are carcinogens, like, oh, if you smoke cigarettes, that's a carcinogen. A carcinogen is simply a mutagen that causes cancer. And carcinogens are everywhere in our common society. So carcinogens are in things like cigarettes, which we know, but they're also in your produce. So one of the most heavily used chemicals in the world is an agricultural chemical that kills weeds. It's increased our food production, but is it having a negative consequence? Some people suggest that because Roundup is a human carcinogen, that it might be tied to an increase in the rate of cancer because when we don't buy organic vegetables and Roundup is used, maybe we're exposing ourselves to carcinogens. Now, this is still some studies going on, and I can't tell you for sure what the outcome is going to be, but it's certainly interesting and something to take a look at. That wraps up our lecture on mutations. Thank you for sticking with me, kiddos.